At this point, you probably would like to know if these molecular traits are somehow associated with a specific biological or clinical feature of the samples. To do this, we can switch to the other environment typical of the genome cruiser, which is the analysis mode. Here, samples are represented as small boxes sticked to the lateral wall on the right, and their clusters are represented in darker gray as bigger objects that form groups. So here we have the transcriptional cluster A and B, and inside them the copy number, low copy number variation cluster and high copy number variation cluster, and within this the methylator phenotype, low, intermediate and high. On the other side, on the floor, we have the features that are used to describe these samples. And this can be genes, and again we have the same cluster we described before. So the methylation cluster A, B and C, and within them the transcriptional subclusters. So for example C3, C2 and C1. Going to single objects, we have genes, so we can look at individual genes by pointing to them. For example, here in expression cluster B2, we have plaque HB1, and also we can have known gene objects or features that describe the samples. And for example, here we try to include some clinical parameters, which are grouped here. And within them, we have the stage, we have the microsatellite instability status, and the anatomic site for the primary lesion. So we can exploit these features to investigate whether the molecular traits we used to classify the samples have some clinical or biological relevance. And to do this, we can select, for example, the MSI status, and immediately its values are plotted across all patients. And in particular, green patients are endowed with microsatellite stable status, while black patients display a low level of microsatellite instability, and red patients are endowed with a high level of microsatellite instability. And it's immediately evident as red patients are extremely enriched in this subcluster here, which is characterized by transcriptional cluster A, low copy number variation, and high methylator phenotype. The same is true also for the site of the primary lesion. So again, in green we have samples that come from the ascending or right colon or proximal, while the black samples are coming from the transverse colon, and the red samples come from the left or distal colon. And again, here we can see two relevant features. The first is that samples from the left colon are extremely enriched in this sub-cluster here, while the second feature is that samples from the right colon are extremely correlated with the high methylator phenotype, as you can see here. And in this case, even if the molecular trait is scattered around the different sample clusters, in any case it's clear the correlation because of the color code used to define the feature. If we look at the stage, again we can see its values around here, and in this case stage 1 samples are in bright green, while in dark green we have the stage 2, in dark red stage 3, and in bright red stage 4 samples. And even if in this case, as expected, the values are more scattered around, again we can see a clear enrichment of stage 3 and stage 4 tumors in this subcluster here, which is the intermediate methylator high copy number class B transcription. Now, possibly you would be interested in identifying the key and the specific molecular feature that can characterize this subcluster of patients. To do this, we can just select this subcluster of patients and the opposite applies. So immediately the average value within this number of samples is exploited for each gene in our dataset, and the same is true also for the clinical parameters. So if we look at single genes, we can see that they have, as before, are endowed with a color, which represents their expression value in this case, and is the average expression within the samples, but they also have a height, are endowed with a height, that in this case is a statistical measure that indicates how significant is the differential expression of this gene within this cluster of patients with respect to the general population. So based on this, it's immediately evident that we have 3 independent gene clusters that are heavily downregulated in this subset of patients. And to get some insights into the properties of these gene clusters, we can use enrichment statistics. And we can exploit to do this the other wall, the front wall, which is called the gene groups wall. Within this wall here, we have a number of buttons. Each of them represent a gene set. In this case, gene sets have been downloaded from the molecular signature database of the Broad Institute. And each box, each button is endowed with a color, a size, and a transparency. The color represents the average expression value of the gene group members within the patients that have been selected, while the size and the transparency depend on the interaction of the user with the floor. So if we select a gene expression cluster, then some groups become very big, others shrink, and some groups are very solid while others are transparent. And this depends on the enrichment statistics that are calculated. In particular, bigger groups are those for which the members are more represented than expected within the gene cluster selection. For example, here we have 6 times more than expected objects represented within the cluster B4. The transparency is inversely proportional to the significance of this enrichment. So, based on hypergeometric calculation, for example, this cluster here is enriched for genes pertaining to the interferon response, here, interferon beta, and also interferon gamma, and for STAT3 targets. So we can end up with the idea that possibly this downregulation seen in these patients leads or is linked to a reduction of the interferon gamma response. And if we select one of the gene groups, now their members are directly selected on the floor. This allows us to confirm that this specific gene set is underexpressed in our patients of interest. But we can check also how this is reflected to the methylation phenotype of these genes, because hypermethylation could be a reason for underexpression. So to do this, we can switch to the methylation dataset. And now we can confirm that in most of the patients from this subcluster here, these genes are hypermethylated, raising the hypothesis that the interferon response in these subclasses of patients is reduced due to hypermethylation of their members. As you can see, this is true not only for this subcluster of patients, but also for others scattered around. And if we look at these ones to see which are them, we can immediately correlate this phenotype with the intermediate methylator phenotype of the patients. So at the end, we can try to co-select all the intermediate methylator patient to see their methylator profile, confirming indeed that Class B genes, to which most of the interferon responsive genes pertain, are highly hypermethylated in this subclass of patients. On the contrary, if we select the high methylator phenotype, we can clearly see that their methylation pattern is complementary and essentially pertains to genes of the class A. So the segregation of the methylation pattern is quite evident among patients and leaves out the class C genes for which no specific methylation can be found.